Hello, everybody. Welcome to the science class. Today is April 8th, 2020. We're still with the remote learning classes. And the topic of the class today is meiosis. The idea is to differentiate what meiosis is compared to mitosis. If you can remember in our last class, or the project, the hands-on project that we have done with the cancer activity in the WISE platform, we studied mitosis as a cell division process where you have to make exact copy of the DNA inside the cells. So the cells they make exact copies of the chromosomes, right? In a process we can call also as cloning. Well, today meiosis, we try to identify the features that make this process unique because it's something that we're gonna make the sexual cells, in other words, the sperms and the eggs. So let's take a look at it in a deeper way. Um, so in this presentation, we talk about the process of meiosis as a process that represents the sexual reproduction. So meiosis is a production of an offspring as a result of combination of genetic materials from two different cells. Remember, we have to combine an egg with the sperm in a process we call fertilization, and that's the only way you can have a sexual reproduction. So this is not cloning at all. This is not mitosis. The two sex cells responsible for the sexual reproduction are, again, sperm that comes from the female part, and the sperm that comes from the male part, and the egg that comes from the female part. When they combine together, we have a process we call fertilization. Definition: it's a process by which sperm and egg fuse together. After fertilization, the sperm or egg combination is called a zygote. All right. So this is a picture. It's a microscopic picture, electronic microscopic picture of the, the, um, the egg surrounded by many different um, sperms. This must be the human species, but other species they have pretty much very similar shape. And of course, only one sperm is able to break the membrane of the egg. Remember, we're talking about a cell, so this is a cell membrane. So only one sperm is able to break this membrane and get into the egg and fertilize it, making the zygote. So my first question to you is, what's sexual reproduction? If your answer is anything related to fertilization, anything related that the sperm and the eggs combine together to make um, the next generation of the species, you got it right. Another checkpoint is, what are the two sex cells called? I just told you, sperm and eggs. And what's fertilization? That's right. That's a process where you have the eggs and the cells combine together, making the zygote. All right, moving on a little bit. Let's talk about the advantage of the meiosis process. Well, there are basically two different advantages for meiosis. One is what we call the genetic variation. I'm sorry, the genetic variation. So no two individuals are the same. You know that nobody's the same, not even twins are the same. This variation allows species to adapt to environmental traits and evolve. The way that specific eggs with specific sperms combine together, they make unique individuals. And that's the process that makes variation in the nature, variation in the ecosystem. So it represents, for example, that different people respond differently when they are exposed in the environment. Example, the flu virus does not affect every human the same way. Some get very sick while others are barely bothered by it. Nowadays, we're suffering with this COVID-19 situation. It happens exactly the same way. People respond differently to the environment they're exposed to. Another advantage of meiosis is the selective breeding. You can breed certain desirable traits, like farmers and pet breeders have been doing for generations. In the agriculture science, it's very important, for example, for me to have my fruits or vegetables the most beautiful and apparently yummy as possible. So we can breeding. You can try to check for these different traits and focus on the traits that I want the most, which is something we're going to see this in genetics later, which is the next unit. But that's the process of meiosis that naturally allows um, our species of fruits and vegetables to show up like that. Well, this would be impossible if everything was identical cloning, right? If everybody's the same, we don't have any selective breeding. What are the disadvantages of meiosis? First of all, the fertilization. Fertilization process is can be very complicated. Complicated. It's not guaranteed to work. It's not guaranteed that when you're exposing the sperm trying to get into the egg, that process will happen. This fusing process will happen. It's not guaranteed. So that's the first. So organisms created by sexual reproduction usually grow slower. It's a complex mechanism. So of course it takes more time, right? Different from mitosis, that every time that you slap your I'm sorry, every time you slap your skin, some cells die, and very fast by mitosis, by just copying the genetic material, um, you rapidly have the creation of new cells. Okay? Well, if you can list two advantages and two disadvantages of the meiosis process, you're good. Take a look at the Simpsons family. You recognize they are from the same family. You recognize they are from the same human species or Simpson species, but they are all different. They are unique, right? So that's what meiosis does with us. So let's take a closer look at the meiosis process to understand what happens. How meiosis work? Well, you can think of meiosis as a mitosis happening twice in a row, called meiosis 1 and then meiosis 2. The second division is necessary to maintain the right amount of chromosomes. So in the meiosis process, you have to divide the chromosomes to each side of the cell that's created. And when these chromosomes are separated, and then you separate the chromatids, and then you make instead of two, each one of these divided in two again, and you have four in the end. And the idea is to create the gametes, or in other words, the idea is to create the sex cells, either um, uh, eggs or sperms. At the end of meiosis one, when you create the two cells, when one cell becomes two, at the end of this process, you are left with two cells that have 46 chromosomes. So you maintain, you keep with the same number of the original species, right? Just like mitosis does. There is a trick thing that happens here that differentiates from mitosis. I'll tell you in a second. In meiosis 2, when these two cells now start dividing again, two cells from meiosis 2 divide again, resulting now in four cells. So each one of these will become two more, and then a total of four cells, which is the double number compared to mitosis. Each of the four cells in the end of the process now has half of the chromosomes number. They have 20, in the human species, for example, there are 23 chromosomes only. Cells with 46 chromosomes are called diploid. So in the beginning, the original cell is diploid. So your cells in your liver, in your lungs, in your skin, in your body cells, they are diploid. They have 46 chromosomes. However, the cells in your sex cells or your sperms or eggs, they only have 23 chromosomes. They have half of the genetic information because it separates, maintaining the, the original number of chromosomes, and then each one of these will divide, reducing the number of chromosomes by half. And that's what we call haploid, right? So the cells that are sex cells, when we create the sex cells, they are called haploid because they have half of the information, right? Um, Another thing that's important is that the number of chromosomes is 46 for human, but every single species has a different number of chromosomes. For example, I know that a species of rice plant, rice, they have 22 chromosomes. 48 is chicken, and there are others. I think 14 for rats. So the number of chromosomes identify which species you're talking about. We're all constantly working with 46 because that's what humans are about. Okay. All right. So let's go for meiosis one. What happens during the meiosis one? Remember, during the meiosis process, we also have the same phases that happens in mitosis. PMAT, PM AT, prophase, metaphase, anaphase, telophase. Um, the prophase one, during the prophase one, the nuclear membrane disappears, the centrioles go to the edge of the cells, and the spindle fibers are starting to be produced, right? You have a duplication of the pair of chromosomes. Remember, you have a chromosome in blue come from father and another in red come from mom. They are duplicated, make another copy of themselves, right? And if you can take a special look here, take a look at here, how the chromosomes they combine to each other, how they are actually exchanging pieces. That's a process we call crossing over. So crossing over is the process when pieces of chromosomes are being exchanged. And this is the natural process that happens every time we are producing the sex cells, so we can guarantee the uniqueness of life. Or in other words, you can guarantee that we are different from each other. All right. Well, and then you can see in the metaphase. In the metaphase, these chromosomes will be aligned in the middle. Remember, meta, middle. I told you in the last video. The metaphase, the chromosomes are aligned in the middle. The spindle fibers attached to the centromeres, this central part here of the chromosomes, and they are pulled to the edge of the cells towards the centrioles, right? But when the chromosomes are aligned in the middle, we call this metaphase. It's metaphase one because we're still talking about the uh, diploid cells. Anaphase, now you can see the different after crossing over. You see the blue part exchange a piece of red part of the chromosome. The same happens here. The blue one with the red one, the red one with the blue one. So that crossing over guarantees that we are different from each other. All right. The anaphase is when the chromosomes are dragged to the edge of the cells. And finally, the telophase. The the telophase is when the chromosomes are in distinct parts of the cells and cytokinesis starts happening. So you can see already the division of the cell happening here in the middle, and the nuclear membrane starts forming once again. All right. Well, so now we talk about the process of one cell becoming two. So let's try to focus on each of these cells now that are diploid and reducing the number of chromosomes. That's the second phase of meiosis, and we go over all these four different phases again. PMAT, but now prophase two, metaphase two, anaphase two, and telophase two. Now, instead of dividing the chromosomes, we will be dividing the chromatids. So in prophase again, we have this uh, um, uh, nuclear membrane disappearing. Again, the centrioles moving towards the edge of the cell and start making again the um, spindle fibers, right? They already have exchanged piece of DNA during the prophase one in a process we call crossing over. So that's why you see the different colors here in the chromosomes. Metaphase two, you align them in the middle, uh, and once they're aligned in the middle, now the chromatids, not anymore, the chromatids will be pulled towards the edge of the cell. So the chromatids are separated, and in the end, you're gonna have four different cells, and they are haploid. Why are they haploid in the telophase two? Because they only bring half of the information, half of the number of chromosomes. In humans, if you have 46, you in the end you're gonna have 23. Why this number? Well, because later for fertilization, the zygote will be formed, bringing half information from father, 23, half of information from mom, another 23 chromosomes. Combined together, they have 46 chromosomes for the entire individual. And then that's mitosis that copy the types of cells to make the individual grow. During prophase one of meiosis, homologous chromosomes will exchange bits of and pieces of each other. This process is called crossing over and will make each chromosome unique, increasing diversity. That's a summary of what I have just said. This is a very nice picture, microscopic picture, electronic microscopic picture when crossing over happens. Remember, it's all a chemical interaction process. Well, what's the purpose of meiosis? Is to produce gametes and um, guarantee that we're all different from each other. What are diploid cells? Are the cells that carry 46 chromosomes in case of humans, uh, and consequently haploid are the cells that carry 23 chromosomes, again, in our example. And crossing over is a process where you have the exchange of genetic material during prophase one. Well, as a scientist, I'm sorry, we don't have homework here. So this is the end of the video today. I hope you enjoy it. If you go to Google Classroom, there is, for the day of today, April 8th, there is a specific video and a specific assignment for you to complete. I hope you understand. If you have questions, please let me know. See you guys.